pretty sure the weight is normalized by a model. They just check your car model.
Yeah, and I found it in the end. But luckily, it was lighter than I thought it was.
Perfect.
I know. I was quite happy about that. Makes a difference of like 500. It's insane.
500?
Pretty nice.
Kilograms? No, but
Euros.
Euros. For you pay. Okay.
Yeah. Tiny change in weight. 500 euros difference. Anyways, it's 9:30.
Change of topic.
Welcome to Neuroccino. Today we have Hervé present a paper to us that he chose, and we are in the very fortunate position that we have the authors, well, part of the authors in the room to address your questions that you have afterwards. For those of you who are new, Neuroccino is a weekly journal club. We meet every Monday morning, 9:30. Anyone is welcome to join and volunteer themselves to present exciting science, and that can be a book chapter, a paper. We also had YouTube videos. So whatever got you excited about science, do come and talk about it to us. The only rule is that you do not present your own work. And that's about it, I think. So without further ado, if you want to take it away, share the link, please, in the chat so I can copy it on YouTube, and then share the paper in full screen for us, please.
Yes. Okay, let me share the link first. I didn't prepare that, so I need to now to do it.
Oh, you're a Neuroccino veteran. You should know.
Yeah. Okay. Next time. Okay, here is a link.
I like it. I see new faces in the room. That's fantastic.
Okay, so here's a link. So that is down. Okay, so and now I need to share the screen.
That'd be helpful.
Yeah. Yes. So share.
Fantastic.
Okay. You can see it? Yes. Normally technical. Okay. So I'm little, as Stephanie said, I'm little bit stressful because we have maybe for the first time, I don't know, some of the authors of the article. So thanks, Barbara, to be here. So we met at OHBM talking about this topic about looking at the evolution of the human brain using neuroimaging and also genetics. So it's stressful because I need to be careful, not making any mistakes on what I'm describing. But on the other side, I would not. So for the questions, maybe I will have pushed the question to Barbara, and so I will be free of that. Okay, so to start with, so this is the paper using neuroimaging genomics to investigate the evolution of human brain structure. I want to go back to last year, another Neuroccino I was presenting that was about temporal mapping of derived high-frequency gene variants support the mosaic nature of the evolution of Homo sapiens. So I was presenting this paper when it was not already published, and it was about how you can give a date to a polymorphism to a variant. So you can really put the variants in a timeline. So that was really cool. And I did a tremendous work on my presentation, as you can see. But nevertheless, so the point is that I did a slide about how you can study brain evolution of Homo sapiens. So you have different approach. So you have the direct approach, which is looking at the brain of our ancestors. Unfortunately, you cannot really do that because we don't have the brain of these of our ancestors when we go back to a long time. So what we have is, for example, the skulls, fossils of this brain. So you can study them as a proxy these curls or even other part of the body. Another approach is to do comparative study. So you take the brain of current primates that share common ancestor with us, and we look at the differences and similarities to decipher on the evolution of the brain. And the third approach you can use is what I call it paleogenetic brain, which is using genes to look at the evolution of the brain. So we know that the genome keeps tracks of our evolution. So there is a record in our genome how our brain evolved. So that can give us some insight on this evolution. So this paper is really in this topic, looking at these genomes, at the genes to decipher on the evolution of the human brain. So to start with, this paper is a follow-up of a previous paper, which is the one from Tilo in 2021, which was on the same topic, the evolutionary history of common genetic variants influencing human cortical surface here. So I want to show you this paper just for this figure where you can see a little bit more details on the approach. So in the genome, as I said, you have records our evolution. So here you can see a timeline here present, and you can go back to 30 million years with genetics. So you have what we call different genomic annotations you can use. For example, singleton density score, Neanderthal integration. So here I want to just to open a parenthesis. I'm pretty sure you're aware of the recent Nobel Prize Svante Pääbo, who worked on this archaic DNA, and was one of the researchers showing that we interbreeded with Neanderthal, and so we have part of this genome in our genome. So we can look at this annotation to see what is the influence, the effect of these genetics elements on our, for example, like in this paper, on the brain. And so you can go back like that using different annotations, selective sweeps, human accelerated regions, and also human gain answer. So you have this timeline, and you can correlate these genomic annotation with phenotypes like the brain. So what they did in this paper was to look at the cortical metrics like thickness, surface, and volume, and they show that, for example, I will come back to that, but the human gain answer, one of the genomic annotation, is correlated, associated with the variation of cortical surface in different regions. Okay, let's go back to now. So they wanted to do follow-up. One of the main reasons is that in the previous paper that was part of the Enigma effort, so when you do that, you agglomerate different cohorts of subjects to get the number, the sample size enough to conduct genomic analysis. So, but doing that, so you mix different populations. So you can have what they call population stratification that can bias your results. So the idea here was to not use different population, but to use only one, the UK Biobank dataset, which now is pretty big. And for this study, they use 30,000 subjects. The aims, as I said, the aims of this paper is, there is different aims. First of all, replication, and also to do new analysis. So we'll go in details on that. So they wanted to look at both hemispheres because the previous paper was only was not interested in asymmetry results, using new annotation and also using new metrics. Because in this paper, they will use the cortical metrics, but also diffusion MRI metrics. Okay. So they use, so just to recap, 30,000 subjects from UK Biobank, looking at cortical metrics and diffusion white matter metrics. And using the same or similar annotation I showed you before. Let's go back to that. So these different annotations. So like that, you can correlate different time point in time. Okay. So first of all, so they wanted to see if their analysis, so if it is replicable. Here you can see on this figure the genome-wide association analysis for the previous sample, the Enigma sample, and for the UK Biobank replication sample. As you can see, so that is the genome-wide, the GWAS for the cortical surface, the average. And so you can see it's pretty similar. After that, they wanted to replicate their results. So they selected the regions they found significant in the previous study, and they redid the same analysis on. So first of all, they didn't replicate one of the results, which was on the single atom density score, which is a method for detecting sign of recent polygenic selection. So if we go back to so this is this one, the most recent annotation. So they didn't find back these results, and mostly due to maybe this stratification, population stratification on the previous study. But what they found is on the human gain answer, the association with cortical surface on the pars orbitalis region and the human gain answer annotation. Okay, so and yeah, so to go back to this human gain answer annotation, so here we are at the bottom of the timeline, so around 30 million years, and these genetic elements. So these annotation give insight on genetic element that gain activity in the human. So they compare expression of these annotation in macaque, human, and mouse, and comparing these expression, they found that these genetic annotation was specific to human. Okay, so that was their replication. As I say, they wanted to do more, so they redid also. The first figure was a replication, so they removed. So as in the Enigma sample, they got some subjects from the UK Biobank. So they removed from the 30,000 the subjects that belong to the previous sample. So, but for the next results, they pull everybody together. So these results are now on 30,000 subjects. So they wanted to look at the effect on both hemispheres, so separating left and right. And so here you can see the association between, again, the human gain answer and cortical metrics, and here specifically cortical surface. So you can see that you have the left pars triangularis, the left pars opicularis, the supramarginal, and you have some of the occipital regions on the left and the right. So you can see that what is interesting in these results, we have some regions that are really implicated in language, so that can make stories about it, and we have also regions in vision function. Okay. So as I say, so that was on cortical metrics. After that, they use in UK Biobank, you can also have the diffusion matrix. So here they look at the different diffusion matrix. Here is the fractional anisotropy. And what they found is a depletion for the Neanderthal integration elements for the uncinate fasciculus. So depletion, so what I showed before was a positive correlation, saying that the more you have the elements, the more it's associated with brain phenotypes. Here is the opposite. The more you have these annotations, the less it's associated with the phenotypes, and here specifically the uncinate fasciculus. Okay. So after that, they wanted to look a little bit more on the pars, so the results on the pars triangularis region on the left, and they wanted to see. So they did so when you do this type of analysis, it's like if you do a genome-wide association. So they wanted to see specifically what were the polymorphism, the variants that were implicated in the variation on the surface on the pars triangularis. So they did that. They found different hits. So you can see here the different SNPs variants that are associated. But they wanted to see the SNPs that are specifically in the human gain answer annotation. So they wanted to have an overlap between the polymorphism that are significant and that belong to the human gain answer. They found one SNP in particular, these RS1875748, that was significant and within your human gain answer and located in a gene which is ZIG4, gene implicated in the neurogenesis during development. So what is cool about genetics, they have plenty of tools to explore their results. So first of all, you can select your polymorphism and see, yeah, so it's implicated in ZIG4. So I want to see what is the effect of the different alleles on the expression in the brain, in the human brain. And so you can do that. You have tools to do that. So when you select these SNP, you can see that when you have TT, so the polymorphism is a T or C. You can have CC, CT, or TT. And when you have TT, you have more expression of the ZIG4 protein, not protein, mRNA in the brain compared to CC and CT. What is interesting is that the T allele is the archaic allele. So you can see that the primates that share a common ancestor don't have these new C allele. Only Homo sapiens, Neanderthal, and Denisovans have these polymorphism. So as I said, they have plenty of tools, and one of these tools is, and now we go back to the paper I just showed you at the beginning, looking at the date of the polymorphism. So there is now a web site where you can put the polymorphism you are interested in, and it gives you when this polymorphism was created. Let's say that like that. And they found that if we go back to this, so it's around 46,000 generations. That makes around 1.2 million years. Okay, so we go back in time. So that is pretty cool. So we know that it's estimated that these variants happen at this time. So, okay. So that is all the results. I think there is some limitation they discuss in the paper. So first of all, they use diffusion metrics, but which are very basic. So I think, yeah, that will be helpful to use other metrics like coming from tractography and stuff like that. It will be very interesting. They use only European population. That was the point. But yeah, using only one population, you cannot generalize. And I think it's more general limitations that with this kind of approach, we look at polymorphisms that are still variants. And so we know that a lot of these polymorphisms are now fixed in the current living population. So we cannot study them and their effect on our evolution. And something we need to also to take into consideration is that these annotations are roughly in this panel, but that they will move. We need more annotation, more details on these annotation, but that is for the future. So I think I will stop here. That this study gave clues on and give insight on research pathway to conduct new study on this evolution and combining neuroimaging and genetics. And for those who want more, there is a very nice trade by Simon Fisher on this paper that give you a real summary on the results. And that's it for me.
Lovely, thank you, Hervé. As always, you take us far beyond the paper. I love it, and I'm learning a lot. Are there any questions at the moment? I saw Barbara taking notes in between, so I guess she has something to say about it as well.
I made mistakes. Oh no.
Probably more comments. No questions? Okay, in that case, let me go first. Also, for those watching us over on YouTube, you're welcome to write your question in the chat over there, and I'll bring it into the Zoom live. So I was quite intrigued by the results with the white matter. If you could go back to the figure with the uncinate. Here we go. So I'm quite puzzled by it because the uncinate is quite a lateral connection, right? I mean, it goes through the temporal stem, but other than that, it's connecting quite new areas of the brain. So I was surprised to see that one is coming up as the significant connection and not a white matter structure that is deeper in the brain and the evolutionary older parts. So I was just wondering if any of you presenter or authors might want to comment on that.
Barbara, you want to say something?
I love how you just give the responsibility.
I think I'm also looking a bit at my student because the first author is here as well. So okay. I think it's not that you are just the one that is a bit puzzled. I think we were as well. I mean, on one side, it is nice that it's something that is in some form related to language, but on the other side, there is also, I think it's one of the most stable tracks that you can find and not something relatively new. And then on top of it, it has a depletion of Neanderthal introgression. So wrapping your head around this was a bit complicated. I think we gave a few hints of how to interpret it, but it's not the easiest finding. Yeah.
Let me just apologize. I didn't see the first author because they just changed the name halfway through.
No worries.
No.
Hiding.
Everyone, sorry.
Well, thanks for joining us.
Yeah.
It was a great presentation. Thanks again. About the depletion for uncinate fasciculus, maybe a quick comment from my side could be that, so this finding is specifically pointing to differences between Neanderthals and humans, I guess, or like Homo sapiens, which is supposed to cover genetic differences which came out in last 500,000 years or so. So in that sense, the neuroanatomical differences between Neanderthals and Homo sapiens are sort of mostly unknown, I guess. And that's why I think it's a bit difficult to interpret these depletion, or if we would have find any enrichment as well, that would also be kind of difficult to interpret anyway. But for the white matter tracts that have evolved in older times, for example, the ones which are different between humans and macaques, let's say, then I think we would expect to see an enrichment or a depletion within the human gained enhancers, for example. But for this annotation, at least in my mind, it kind of makes sense that white matter structures that evolved more recently are showing up as significant. So maybe that's...
And then, as Hervé also mentioned during the presentation, I guess the measurement was FA based only, right?
Mm-hmm.
Yeah.
Mm-hmm.
Okay.
That would be good to have more fine metrics to look at that, I think.
Yeah, but I mean, in the end, that's like the one thing. You're limited with what UK Biobank gives you because you need summary statistics. So that's it. That was what we have and what we tried. And I think what's the most common, but definitely we're, I think, always looking at better metrics to test what we want to test.
The way to go. Another question from Chris.
Hi, thank you for this very interesting presentation. I don't know I pretty much don't know anything about genomics, but what is the time frame that you can observe changes with this technique? Can you differentiate what could be due to modern lifestyle, or is it just between humans and other like Neanderthal and other I don't remember the word for the other not species, because we clearly mixed with them, but yeah, what's the time frame you can look at with this technique?
Yeah, so very briefly again, as it was shown in the presentation very nicely, in the previous paper from Amanda, there's this timeline, and there are different methods which give you an idea about different time frames. For example, the method which is informative about last 2,000-3,000 years is singleton density score. So that might be more relevant to more recent changes in, for example, culture or human lifestyle or population movements around the world and so on. So that might reflect that. But if you're interested in, I think, cortical evolution, most of these traits were fixed probably, let's say, 2 million years ago, or maybe even before in the human lineage. So it's a bit difficult to pick an annotation or a time frame that works well genetically, but also answers questions about, like, evolutionary questions about brain evolution, because it should overlap with your questions. The method should overlap with your questions. And the reason why we didn't use singleton density score in the new paper, or the non-replication part of the new paper, was because it was, I think, a couple of years ago, it was shown that this method is specifically sensitive to population stratification. Even if you try to account for that, there will be some sort of confounder effect probably. So that's why we left it out in the end. And also, given that we think that these traits have evolved in older, like deeper time frames, we focus more on human gain enhancers and Neanderthal introgression parts. So.
Okay, thank you.
Yeah, no problem.
I can ask a non-content related question to you. You're obviously dealing with a gigantic amount of data here, and very complex questions. I was wondering what you personally found the most challenging about this paper and bringing it into existence.
Barbara?
I would definitely say the interpretation of some of the findings, because you don't want to over-interpret what you have and how much impact it has, because in the end, you always are basing things on SNPs that were, for example, significant in the GWAS, but you don't know which is the SNP that actually is causal. So I think having this balance between interpreting and not over-interpreting it and finding ways and to move forward. I think that from like my side was sometimes a bit tricky.
Yeah, I think I agree. Interpretation of the results were probably the most challenging part. But also, I think this type of analyses are just emerging, and use of evolutionary annotations with heritability associated analysis is slowly, I think, becoming more and more popular. So in that sense, at the beginning, implementing all the methods and partitioning heritability with these custom annotations and so on, that was also slightly challenging, but in the end, I think we are quite okay now.
Yeah. I think you spend a long time in like making it more bulletproof and finding annotations that they are stable enough and dealing with a lot of issues that we at some point were like, that makes no sense, what's happening here. But then in the end, I think now we found quite a good way and have a pipeline that I would say runs quite smoothly.
Very nice. Well, congratulations on this fantastic study. Thank you, Hervé, for presenting despite the authors being present.
Welcome.
Thank you very much to the authors for joining us. That was fantastic having your input straight away today. So thank you very much for taking the time. Thanks everyone else for being here. This is all the time we've got this morning, I'm afraid. So I have to let you go and wish you an exciting week in science. And see you next Monday morning, bright and early, for a new Neuroccino. And I think next week we have Victor present, or Teresa, one of you. Victor. Cool. Fantastic. See you next week. Bye.
Bye.